Adequate kidney function is essential to the maintenance of a healthy body. If there is complete kidney failure and treatment is not provided, death is inevitable. The kidneys are the principal organs of the urinary system. The primary functions of the kidneys are to regulate the volume and composition of extracellular fluids and to excrete waste products from the body. The kidneys also function to control blood pressure, produce erythropoietin, activate vitamin D, and regulate acid-base balance. Urine is formed in the kidneys, drains through the ureters to be stored in the bladder, and then passes out of the body through the urethra. The paired kidneys are bean-shaped organs located retroperitoneally, which means behind the peritoneum, on either side of the vertebral column at about the level of the 12th thoracic vertebra to the 3rd lumbar vertebra. The right kidney, positioned at the level of the 12th rib, is lower than the left. A thin, smooth layer of fibrous membrane called the capsule covers the surface of each kidney. The capsule protects the kidney and serves as a shock absorber when this area is traumatized from sudden force or strike. The hilus on the medial side of the kidney serves as the entry site for the renal artery and nerves and as the exit site for the renal vein and ureter. The parenchyma, actual tissue of the kidney, can be visualized on a longitudinal section of the kidney. The outer layer is the cortex and the inner layer is the medulla. The medulla consists of a number of pyramids. The apices, or the tops of these pyramids, are called the papillae, through which the urine passes to enter the calyces. The minor calyces widen and merge to form major calyces, which form a funnel-shaped sac called the renal pelvis. The minor and major calyces transport urine to the renal pelvis, from which it drains through the ureters to the bladder. The renal pelvis can store a small amount of urine, about 3 to 5 milliliters. Each kidney contains approximately 1 million nephrons. Each nephron is composed of the glomerulus, Bowman's capsule, and a tubular system. The tubular system consists of the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting tubules. The glomerulus, Bowman's capsule, proximal tubule, and distal tubule are located in the cortex of the kidney. The loop of Henle and the collecting tubules are located in the medulla. Several collecting tubules join to form a single collecting duct. The collecting ducts eventually merge into a pyramid that empties via the papilla into a minor calyx. The blood supply to the kidneys is about 1,200 milliliters per minute, or 25% of the total cardiac output. Blood flows from the aorta through the hilus via the renal artery. The renal artery branches into the afferent arteriole. Afferent arteriole branches into capillaries. Capillaries turf to form the glomerulus. Capillaries reunite to form the efferent arterioles. Efferent arteriole splits into the capillary network called the peritubular capillaries. Peritubular capillaries drain into the venous system. The renal vein empties into the inferior vena cava. Urine formation is the outcome of a complex multi-step process of filtration, reabsorption, secretion, and excretion of water, electrolytes, and metabolic waste products. Although urine formation is the result of this process, the primary function of the kidneys are to filter the blood and maintain the body's internal homeostasis. Filtration and reabsorption occur via hydrostatic pressure and osmotic pressure. Hydrostatic pressure is the force of fluid in a compartment pushing against a cell membrane or vessel wall. In the blood vessels, hydrostatic pressure is the blood pressure generated by the contraction of the heart. Hydrostatic pressure in the vascular system gradually decreases as the blood moves through the arteries until it's about 30 millimeters of mercury in the capillary bed. At the capillary level, hydrostatic pressure is the major force that pushes water out of the vascular system and into the interstitial space. Osmotic pressure is the concentration of the solution which determines the strength of the osmotic pull. The higher the concentration, the greater a solution's pull or osmotic pressure. Urine formation begins at the glomerulus where blood is filtered. The glomerulus is a semi-permeable membrane that allows filtration. The hydrostatic pressure of the blood within the glomerular capillaries causes a portion of the blood to be filtered across the semi-permeable membrane into Bowman's capsule, where the filtered portion of the blood, which is called the glomerular filtrate, begins to pass down to the tubule. Filtration is more rapid in the glomerulus than in ordinary tissue capillaries because the glomerular membrane is porous. The ultrafiltrate is similar in composition to blood except that it lacks blood cells, platelets, and large plasma proteins. Under normal conditions, the capillary pores are too small to allow loss of these large blood components. However, in many kidney diseases, capillary permeability is increased, which permits plasma proteins and blood cells to pass into the urine. The amount of blood filtered each minute by the glomeruli is expressed as the glomerular filtration rate, or the GFR. The normal GFR is about 125 mLs per minute. The peritubular capillary network reabsorbs most of the glomerular filtrate before it reaches the end of the collecting duct. Therefore, only about 1 milliliter per minute, on average, is excreted as urine. Since the glomerular membrane is a selective filtration membrane that filters primarily by size, provision is made for the reabsorption of essential materials and excretion of non-essential ones. The tubules and collecting ducts carry out these functions by means of reabsorption and secretion. Reabsorption is the passage of a substance from the lumen of the tubules through the tubule cells and into the capillaries. This process involves both active and passive transport mechanisms. Tubular secretion is the passage of a substance from the capillaries through the tubular cells into the lumen of the tubule. Reabsorption and secretion cause numerous changes in the composition of the glomerular filtrate as it moves through the entire length of the tubule. In the proximal convoluted tubule, about 80% of the electrolytes are reabsorbed. Normally, all the glucose, amino acids, and small proteins are reabsorbed. As reabsorption continues in the loop of Henle, water is conserved, which is important for concentration of the filtrate. The descending loop is permeable to water and moderately permeable to sodium, urea, and other solutes. In the ascending limb, chloride ions are actively reabsorbed, followed by passive reabsorption of sodium ions. About 25% of the filtered sodium is reabsorbed in the ascending limb. Two important functions of the distal convoluted tubules are the final regulation of water balance and acid-base balance. Antidiuretic hormone, or ADH, is required for water reabsorption in the kidney and is important in water balance. ADH makes the distal convoluted tubules and the collecting ducts permeable to water. This allows water to be reabsorbed into the peritubular capillaries and eventually return to the circulation. Decreases in plasma osmolarity are detected in the anterior hypothalamus by osmoreceptors. These osmoreceptors send neural input to the supraoptic nuclei cells in the hypothalamus. These supraoptic nuclei cells have neuronal axons that terminate in the posterior pituitary gland and act to inhibit secretion of ADH. In the absence of ADH, the tubules are essentially impermeable to water. Thus, any water in the tubules leaves the body as urine. Aldosterone, released from the adrenal cortex, acts on the distal tubules to cause reabsorption of sodium and water. In exchange for sodium, potassium ions are excreted. The secretion of aldosterone is influenced by both circulating blood volume and plasma concentrations of sodium and potassium. The renal tubules are involved in calcium balance. Parathyroid hormone, or PTH, is released from the parathyroid gland in response to low serum calcium levels. PTH maintains serum calcium levels by causing an increase in tubular reabsorption of calcium ions and decreased tubular reabsorption of phosphate ions. In kidney disease, the effects of PTH may have a major effect on bone metabolism. This slide is just a brief summary of the waste products that are passed from the body in the form of urine. Myocyte cells in the right atrium secrete a hormone, arterial natriuretic peptide, or ANP, in response to arterial distension, which is a result of an increase in plasma volume. ANP acts on the kidneys to increase sodium excretion. At the same time, ANP inhibits renin, ADH, and the action of angiotensin II on the adrenal glands, thereby suppressing aldosterone secretion. These combined effects of ANP result in the production of a large volume of dilute urine. Furthermore, secretion of ANP causes relaxation of the afferent arteriole, thus increasing the GFR. Acid-base regulation involves reabsorbing and conserving most of the bicarbonate and secreting excess hydrogen ions. The distal tubule has different ways to maintain the pH of extracellular fluid within a range of 7.35 to 7.45. The kidneys perform vital functions through the participation in red blood cell or RBC production and blood pressure regulation. Erythropoietin is a hormone produced in the kidneys and secreted in response to hypoxia or decreased renal blood flow. Erythropoietin stimulates RBC production in the bone marrow. A deficiency of erythropoietin occurs in kidney failure, which leads to anemia. Vitamin D is a hormone that can be obtained in the diet or synthesized by the action of ultraviolet radiation on cholesterol in the skin. These forms of vitamin D are inactive and require two more steps to become metabolically active. The first step in activation occurs in the liver. The second step occurs in the kidneys. Active vitamin D is essential for the absorption of calcium from the gastrointestinal tract. The patient with kidney failure, also called renal failure, has a deficiency of the active metabolite of vitamin D and manifests problems of altered calcium and phosphate balance. Renin is important in the regulation of blood pressure. Renin is produced and secreted by the kidney's juxtaglomerular cells. Renin is released into the bloodstream in response to decreased renal perfusion, decreased arterial blood pressure, decreased extracellular fluid, decreased serum sodium concentration, and increased urinary sodium concentration. The plasma protein angiotensinogen from the liver is activated into angiotensin 1 by renin. Angiotensin 1 is subsequently converted to angiotensin 2 by the angiotensin converting enzyme or the ACE enzyme. ACE is located on the inner surface of all blood vessels, with particularly high levels in the vessels of the lungs. Angiotensin 2 stimulates the release of aldosterone from the adrenal cortex, which causes sodium and water retention, leading to increased intercellular fluid volume. Angiotensin 2 also causes increased peripheral vasoconstriction. Release of renin is inhibited by an elevation in blood pressure. Excessive renin production caused by impaired renal perfusion may be a contributing factor in the etiology of hypertension. This is a visual diagram of the renin-angiotensin-aldosterone system process I just described. The next three slides provide the diagnostic tests used to determine renal pathophysiology. All of these values reflect normal or abnormal kidney function. Urine-specific gravity and the nature of the solutes found in urine reflect the filtration occurring in the kidneys. Urine output is a vital factor in understanding and determining the function of the kidneys.